Like, hey guys, welcome to my vlog. So, today we're gonna look at enzymes and digestion. Oh my god, how exciting. So, I don't really know why I'm talking like this, but sure, here we are. So, enzymes and digestion. Now, I think we'll split this up into two parts. So we'll do enzymes, which is the bit that's going to take the longest, and then we'll do digestion, and we'll do that in a separate video. Okay, lovely, how exciting. Maybe even Big Alfie will help me out here. Alfie's my dog, PS. So, let's have a look at enzymes. Now, we're going to rattle through this at the speed of light, and we're going to pick out the most important bits here, okay? So, what is an enzyme? An enzyme is one of these. It is a protein which speeds up chemical reactions. And because it speeds up chemical reactions, it's sometimes known as a biological catalyst. Okay, so out of all this, the most important bits are that enzymes are chemical compounds made up of protein, and they speed up the rate of reactions, and they're often called biological catalysts. But this bit here is it summarised in a little bullet points. Now, we've got two types of enzymes. Ones which can break down large molecules into small ones, and these are known as catabolic enzymes. Or we've got anabolic enzymes, which build up large molecules from small ones. So, basically, let's say that we have got some small molecules here, and then we've got some large molecules here. So, let's say that these are small individual molecules, and this is one big large molecules. What we can either do is build them up from small molecules into one big large one, or break them down from large ones into small ones. Now, if we're building them up, we use anabolic enzymes, but if we're breaking down the large molecules into small ones, those are catabolic enzymes. Now, these are the ones which we're going to look at most in this topic, catabolic enzymes, because we're usually looking at proteins being broken up into amino acids, or we're looking at starch being broken down into glucose or sugars. Okay, so most of the enzymes that we look at are catabolic. Now, the first one we're going to look at is a catabolic enzyme, and it's called catalase. Now, this speeds down up the breakdown of hydrogen peroxide. It's a poisonous substance found in the liver, it's a poisonous substance formed in cells, and you find this enzyme catalase in the liver. Now, when you break it up, so hydrogen peroxide is produced in your cells, catalase breaks it down, we get water and oxygen, and that's pretty much all we need to know. We've got a few other wee questions on it. The only other thing that you want to take away is that this is a catabolic enzyme. Now, that's not so bad, is it? Not too bad. Now, this bit is how enzymes work. Now, again, I'm going to keep this really straightforward. So, the molecule on which an enzyme acts is called a substrate, while the molecule which is formed by the reaction is called a product. So basically, we start with substrates. They get built up or broken down into products. And it is the enzyme that carries out that reaction. Now, this bit is really important, and we're going to talk about this quite a lot in this topic. But enzymes work because the shape of the substrate matches exactly the shape of a special region in the enzyme called the active site. Now, this bit, the way it matches exactly, we say that they are complementary. Now, let me draw you a wee picture, and we'll use all these little things in bold. So, let's say, for example, we've got a little enzyme. We'll draw it like a little Pac-Man. Oh man. So here's our little enzyme. Isn't it beautiful? Now, what's going to happen is we're going to have a little substrate, and this is a molecule which is going to get broken down in this case. Now, what you'll notice is the shape of this substrate matches the shape of this area, and this area is known as the active site. So what happens is the active site, or sorry, the substrate can sneak in there and it'll fit its shape perfectly, and what happens is once it goes in, it gets broken down and we end up with products. So these two here, little triangles, are the products. Now what you'll notice about this is that the enzyme hasn't changed. Okay, so we've got the substrate, which is the thing or the molecule that's going to get broken down. We've got the enzyme, which is a protein which speeds up reaction. We said it has a special area called an active site. The active site and the substrate are complementary in shape. And then when the enzyme breaks it down, we get products. Yay, how exciting. Oosh. So the next little bit is all about enzyme specificity. Now, this basically means that each enzyme can only break down or work on certain substrates. Okay, so for example, I've got 3 keys on my little car key, what do you call it? I don't even know what you call it, bundle of keys. But my backdoor key is a little Darth Vader key, so he looks a bit like this with his wee grumpy face and his wee triangle and then all his deep breathing and everything. So the thing about that is that only the little Darth Vader key will open my back door. So like we say that each enzyme can only break down certain substrates, we call this the lock and key theory. So in this case, the key is the substrate and the lock is the enzyme. So the way only certain keys, like for example this one, fit into certain locks, only certain substrates can fit into certain active sites. Okay, and that is because they have complementary shape. So we say that enzymes are substrate specific. Okay, and that's really important. The lock and key theory is all about how enzymes are substrate specific. Now, this shows how they're specific. So each enzyme here will only break down a certain substrate and they'll only produce a certain product. So we've got 4 here, 4 enzymes which we need to know about. Carbohydrase, amylase, protease is going to be this one, and then lipase. Now you'll notice here they all end in ASE. So in biology that usually tells us that these are enzymes. And the other thing is, the first part of it sometimes tells you what they might break down. So we've got carbohydrase, amylase, protease and lipase. Now, there's 1 or 2 which are really obvious. So carbohydrase breaks down carbohydrates. Amylase breaks down starch. Now, the reason it's called amylase is because starch is actually made of something called amylose, but you don't need to know that. We just call it starch. But it explains why it's called amylase and it breaks down starch. Protease breaks down protein, and then lipase breaks down lipids or fats. It is blowing an absolute gale outside. Did you hear that? So stormy. So what are these products that they make? Carbohydrates get broken up into simple sugars or glucose. Amylase gets broken down, amylase breaks down starch, again into simple sugars or glucose. Protease breaks up protein into amino acids. And then lipase breaks down lipids or fats into fatty acids and glycerol. Lovely. Now, these are all catabolic enzymes because they break down large molecules into smaller ones. Now, the next wee bit is explaining why starch can only be broken down by amylase and not lipase or protease. Now, here's how you would answer this. You need to say that enzymes are substrate specific, which means that enzymes are substrate specific, so only starch will fit in the active site of amylase. And this is because starch and the active site have a complementary shape. Now, if we wanted to add more detail, we could say that starch can't fit into the active site of lipase or protease. And that's how that happens. Now, I'll show you a little animation of that, which is here. So, if we look at this one up here, we can see that this yellow part is the enzyme. Here's a little substrate, that doesn't fit. There's another substrate, that one doesn't fit. But OMG, this one does fit. And that is because enzymes are substrate specific. Okay? Now, next little bit. No, thank you. Next little bit is the lock and key summary. Okay? Now, if you ever get asked to describe the lock and key theory, these are the main bits. You need to say, once again, enzymes are substrate specific, which means that they can only control one kind of reaction. So, for example, amylase can only break down starch. This is known as the lock and key theory, which basically means that the way that only one key can fit in a certain lock, only certain enzymes can fit into an enzyme's active site. Now, that bit there is really important to summarise and learn. Okay? Now, next wee bit is inhibitors. So, in this case, what happens is an inhibitor. Now, an inhibitor is something which stops something else working. Okay? So, in this case, an enzyme inhibitor stops the enzyme working. Now, if we go back to this example, we had our enzyme with our active site and we had a substrate which was complementary to it. Now, there will be a reaction in that one. That one will work. Whereas in this one, let's say for example we've got the enzyme, we've got the substrate, but if we put in a cheeky little inhibitor in there, that's the inhibitor. And what you'll notice here is that the substrate won't fit because the active site is blocked, that means that there will be no reaction. And that's how inhibitors work. Okay, so they stop enzymes working by blocking the active site. Now, next bit is factors affecting enzyme-controlled reactions. Now we've got three: temperature, pH, and the concentration of enzyme. Okay, now before we get into this, there's two important words: optimum, which are the conditions the enzyme works best in. So we can have an optimum temperature for all enzymes, and an optimum pH. As well as that, we have got another word which is denatured, and this is where the enzyme's active site changes shape, and that means that the substrate won't fit into the active site. So they are no longer complementary. So instead of having a shape that matches, they got a shape that don't match, cuz. So here's what we got. Now, this is basically showing what a denatured enzyme looks like. So imagine we're a little substrate. A little substrate could come along, and if it has this shape, it would fit in perfectly. But if we take our little substrate out, so there it is there, it can go in there and that fits perfectly. But imagine, are you ready for this? The temperature gets too high, or the pH gets too high. OMG, what a disaster. Now if that happens, the poor little enzymes get denatured. Now you'll see here that they no longer fit perfectly. Okay, so it can't go into there and it can't get broken down properly. So that means that there's going to be no reaction, or it's going to slow way down. Okay, now two main words: optimum and denatured. So the optimum is the condition it works best in. Denatured is when the active site changes shape and the substrate won't fit in, so there's no reaction which takes place. Now we're going to look at three examples here. Okay, now I'm going to just mark it on this because I think it's easier to do it this way. This is a typical graph for enzymes and temperature. Now what we can see here is that as the temperature increases, the rate of the reaction also increases until we get to here, which is the peak, and this is the optimum temperature, which is where it works best. So the temperature where it works best. Now either side of that, there's a slower reaction. So if we go for this, we could say that at low temperatures there is a slower rate. Now that is because at low temperatures there is less kinetic energy, so there are less collisions between the enzyme and substrate, so the reaction is very, very slow. Whereas here, when we get to the optimum temperature, the reaction is fastest. But then we get to here and we get this big drop. Now, this is because at high temperatures, the enzyme is denatured, which means that the active site changes shape which means that the substrate no longer fits. Okay, so the reaction is either slower or it stops. Okay, now that is enzymes and temperature. We will get loads and loads of crazy experiments, but it is all based on that. If we've got low temperatures and the reaction's not happening, that's what we say for low temperatures. Where the peak is highest, or the reaction is fastest, that's the optimum temperature. At high temperatures, the reaction is also very slow, and we need to say these things. The enzyme gets denatured, so the active site changes shape, substrate no longer fits, so the reaction is slower or it stops. Now just on that, sometimes before experiments they say that an enzyme has been boiled. Now sometimes it'll say boiled and cooled. If it's been boiled, that means it's been denatured before the experiment and then it's been cooled down again. Now the bad thing is, say if you're a cheeky wee enzyme, just going about your business, loving life, but then you get boiled, what's going to happen is your active site is going to change shape and the substrates are no longer going to fit, so that means that the enzyme won't work. Now, you can't denature an enzyme. So if this has been denatured, you can't go back in time. Once it's denatured forever. Now, this experiment, oh, what a nightmare. I can't even bring myself to do it. Okay, so, tell you what, we are going to leg it past this. Apart from this wee question, so at the start of the experiment, amylase and starch were placed in the water baths for 5 minutes before being mixed. Why do you do that? So basically we had a test tube of amylase, a test tube of starch, and we did not mix them until they'd been in the water bath for 5 minutes. That is to allow the mixtures or the solutions to reach the same temperature. Okay? Now, the next bit, do you know what? We may as well do this actually. So, and do you know what? We don't even need to look and see what the experiment was. We can just apply what we've talked about in that last wee bit. Explain the results between 10 and 20 degrees C. Well, it's a low temperature, so there's going to be a slow rate of reaction, and that is because there's less kinetic energy, so there's going to be less collisions. So there's going to be a slower rate. Now, then we get the optimum temperature. So say for example we had a graph of results, we would find the peak, and that is our optimum. How could the optimum temperature be determined more accurately? You basically just have to find out what the optimum is and then repeat it more. Now, next one, after 50 degrees C, well, holy moly, that's a high temperature. So that means high temperature, and again it's the same bullet points. The enzymes get denatured and the active site changes shape, so substrate won't fit. So in this case the starch is not broken down. Now, these three never change. It's always the same. The only bit that you might have to include is something which is specific to the experiment. Okay? So, that is enzymes and temperature. The next bit is pH and enzymes. Now, you might remember pH, there's a scale and it goes from 1 to 14. Okay, now, in case you don't do chemistry, 1 to 6 is acid, 7 is neutral, and then 8 to 14 is alkaline. Okay, now, just like every enzyme had its own optimum temperature, each enzyme has an optimum pH. And either side of the optimum, they won't work as well because the incorrect pH changes the shape of the enzyme's active site. Or in other words, it denatures it. So, if we look at this, here we have pepsin. Oh, flip me. Here we have got pepsin and trypsin, and what it says about these are two proteases, so they will break down protein. Now, if we just look at where their highest point is and read down from it, this will tell us the optimum pH. So it's just exactly the same. The highest point is the optimum, and either side it is going to be denatured. It's a bit of a thick pen, but sure. So, if we want to get the optimum pH for each of these enzymes, pepsin has an optimum of 2.5, whereas trypsin has an optimum of 8.5. Okay? Now, either side of that, we can see that the rate of digestion is much, much lower. So, if we look at this, what is meant by optimum pH? It is pH where the enzyme works best. Gave the optimum pH for trypsin is 8.5. Now, this one caused us a lot of problems whenever we were doing this in class. Which enzyme works best in the stomach? Now, in the stomach, there is hydrochloric acid. Now, because it's an acid, it has got a low pH and it has a pH of about 1 to 3. Now, if we look at this graph, pepsin will work really well between the pHs. Well, it will work between the pHs 1 and 5. It works best around 2 or 3. Whereas trypsin will only really work between 7, 8 and 9. Okay, so out of these, it is going to be pepsin, and that is because of the hydrochloric acid in the stomach. Okay, so pepsin, it works best in acidic conditions. Now, explain what would happen if amylase was used instead of protease. Well, this is just exactly what we've talked about, and we say that enzymes are substrate specific. Now, that's because amylase breaks down starch, protease breaks down proteins. So, enzymes are substrate specific. Amylase can only break down starch. So, this reaction wouldn't. In fact, you know what, let's word it slightly differently. Enzymes are substrate specific. Amylase can only break down starch. So, this investigation wouldn't work, or there would be no reaction. And again, that is because enzymes are substrate specific, so amylase cannot break down protein. We are flying through this. 14 pages, OMG. Hey, Alfie, look. Now, next one is concentration of enzymes. So, concentration is like this. Now, we could have a high concentration, high conc, low conc. Now, let's say, for example, we have got the same volume in each one. If we draw in enzymes, which are these little things here, and then we'll draw in a low concentration of enzymes here. If we've got a high concentration of enzymes, that means that we have more enzymes in that certain amount. So because there's more enzymes, there's more active sites. Whereas if we've got a low concentration, we have got less enzymes, so we have got less active sites. So higher concentration equals more enzymes, so more active sites. Whereas if we've got a low concentration, we've got less enzymes, so less active sites. So basically what's happening here is, imagine we are digesting milk protein using probably a protease enzyme because we say it's milk protein. What happens is we increase the enzyme concentration as we go down. So as we're going down, there are more active sites. So more enzymes, so more active sites. Now what you can see here is that as enzyme concentration increases, the time taken to digest the milk protein decreases. The time taken to digest milk decreases. Now, you've got to plot the results on the graph paper provided. We're not going to do that because that would kill everybody of boredom if we did that. So basically what happens is we've got enzyme concentration here, and we've got time taken to digest the milk here. And what we would find out is that as we increase enzyme concentration, in fact I'm going to draw that again. As we increase enzyme concentration, it takes less time to digest it, and then we start to flatten out near the end. So if we're looking at this, there's basically two parts to this, okay? There is this bit where it's really, really steep, and then there's this bit where it levels off, okay? So we'll call this part 1 and part 2. Now, at part 1, what happens is, as enzyme concentration increases, the rate of reaction increases. Now, that is because if we increase the enzyme concentration, there are more active sites. So that basically means that if there is more active sites, there will be a faster rate of reaction. So in other words, the substrates are broken up quicker. Now, at 2, the rate slows down, and that is because even though we're increasing enzyme concentration, there is not enough substrate. So, if there's not enough substrate, the rate of reaction slows down, and we would actually say here that the substrate concentration is limiting. Now, just on this type of reaction, sorry, by the way, this pretty much applies for any graph where we're looking at enzyme concentration. So I know that the time taken goes down here, but what about if it was this way, where it was enzyme concentration and it might be the rate of reaction here? What could happen is, as the enzyme concentration increases, rate of reaction increases, but then it starts to level off. It's the same thing. So as enzyme concentration increases, rate of reaction increases, and that's because there's more active sites free, so there's a faster rate of reaction. But when we get to 2 here, rate slows down because there's not enough substrate, so the substrate concentration is limiting. Now, let's say we were doing a little experiment where we were measuring enzyme concentration and the effect it had on the rate of reaction. How would we make sure it's a fair test? Now, as I said before, we might be using different concentrations. So this would be a high concentration. This would be a low concentration. So what sort of things would we need to keep the same to make it a fair test? We would need to use the same volume of enzymes. We would need to keep the pH the same. We would need to keep the temperature the same. We would also need to keep the concentration of substrate the same. And that's the sort of things that we would need to keep the same to make sure it is a fair test. Okay? Now, last wee bit on enzymes, thank the Lord, is commercial enzymes. Oh my god! Right, so, commercial enzymes. Now, basically, this is all to do with biological washing powders. So if it says bio on your washing powder, it means it contains enzymes. Now, the enzymes that they use are thermostable, which means that they work well in a wide range of temperatures. If that's the case, that's quite good for us, because it means that over a wide range of temperatures, you can clean your clothes and your enzymes will not get denatured. Now, they put them in washing powder so that it can break down food stains. Now, why do they contain a different number of thermostable enzymes? So for example, why do washing powders contain protease and amylase? Again, enzymes are substrate specific. So they can only break down certain substrates. So protease breaks down proteins, amylase breaks down starch, and usually food stains are mixes of different substances. Now, final bit, just before we do that, is this cheeky wee question at the back. Now, this is all about environmentally friendly washing powder. So, if we're looking for an environmentally friendly washing powder, it would have an optimum temperature which was quite low. That means that we need less energy to heat the water. Okay, so that's one of the main reasons why they'll put enzymes into washing powders. It is because if they put enzymes in washing powders, it can break up food stains, especially if they don't dissolve well in water. Now, that was us. I was going to get a little alfie, But I'm actually just thinking here, I think he might be locked out, so I better go and get him. So we'll see if he's okay. But anyway, hope you had fun. I know I did. Well.